Good evening, myself, Dr. Naumita Dhume, and my team, on behalf of Department of Biomedical Sciences, Guru Nanak Khalsa College, Mumbai, heartily welcomes our speaker, Dr. Ajit Datar, and participants from all over India to today's webinar on use of modern analytical instruments in biological sciences. It is said that the great growling engine of change is technology. Technology has reached every nook and corner of the world. After the era of industrializations, systems are getting more better with respect to time consumption, speed, automation, and accuracy. The history of instrumentation can be simply expressed in one word: success. From simple and compound microscopes to now revolutionary scanning and transmission electron microscopes, from demonstrating chromatography on chalk, sophisticated HPTLC, from a basic glass column to modern HPLC, and from the first spectrophotometer in 1940s to the most advanced hyphenated LCMS, analytical instruments have taken a great leap. Modern sample preparations techniques like SPE, SEFE, LME have made it easier to obtain analytes from complex biological matrices, and further analyzing these samples by modern instruments is the need of the hour, where time is becoming a valuable asset. These instruments not only save time but give selective, specific, and reproducible analysis. It is like a one time investment. In the current scenario, RT-PCR is used for diagnosis of COVID-19. To help us understand the use of such modern analytical instruments in biological sciences, who better than Dr. Ajit Datar, our eminent speaker for today? Dr. Ajit Ganesh Datar is currently working as an advisor to Shimadzu Analytical India Private Limited, Mumbai, in the Customer Support Center. His main job profile is to provide instrumental analysis, support to the customers, and provide training. Prior to Shimadzu, he was with Thermo Electron and served this organization as General Manager, Advanced Mass Spectrometry. He was responsible for promoting advanced mass spectrometry products of Thermofinical in India. He retired after completing 50 years, 50 years in 2007. He served for Cromline and IR Technology, Technical Director and Senior Manager, respectively, for a period of 15 years. His first assignment was with Central Research Station of Associated Cement Limited as research officer. He served ACC for 12 years. He has developed methods for characterization of catalysts and processed products using catalysts. He indigenously developed BET surface area analyzer and particle size analyzer. Dr. Datta received his MSc in 1971 and PhD in 1976 from Mumbai University. He was a recipient of Department of Atomic Energy Fellowship in 1971 and carried out PhD research from Analytical Chemistry Division of BARC. He used to work as an adjunct professor and as a PhD guide uh, from Mumbai University. He is currently working as a visiting faculty at several other colleges in Mumbai, including Khalsa College. Thank you, sir, for being here and giving your valuable time for this seminar, rather a webinar. I request Maria to please proceed with the instructions.
A warm welcome to all participants. We request you to follow certain instructions before we begin. Kindly maintain the decorum of the webinar. Avoid posting any irrelevant messages or greetings in the comment section. Limit the use of chat box to only asking questions. The feedback form would be posted in the description box, chat box, and Telegram group after the webinar ends. All registered participants are requested to fill it duly, without which the certificate would not be issued. Now I request Dr. Ajit Datar sir to take over. Thank you.
Yeah, okay. Good evening, all of you. I hope you are getting me very well and you are able to see my screen. Assuming that, I'm proceeding. First of all, let me thank Dr. Nomita Madam for introducing me in nice words, and I welcome you all in such a large number that you are attending this particular presentation, which is on modern analytical instruments in biological sciences. Well, I'm not a biological scientist myself. I'm nobody else other than a person who you are seeing it on the screen. Well, I'm nothing but analytical chemist. I'm a pure and poor analytical chemist playing with some important terminologies in analytical chemistry, and those are sensitivity, selectivity, separation, structure, and speed. Well, analytical chemist has to balance himself while playing with these objects, standing on a platform which has a gyratory motion, and then he has to suppress something, and he has to suppress sampling error, background, and noise so that he can manage to play with these terminologies very well. Let us try to understand these terminologies in slight detail with respect to the biological sciences and applications in biological sciences. Okay, let us go to each term one by one. Sensitivity is the term which is used to find out how low we can measure an analyte in a given matrix. Example could be dioxins in dairy products, meat, etc., and the required level is in femtogram. It requires a GC with high resolution mass spectrometer or very sensitive GC MSMS system. And then other example could be metabolites in vivo studies, especially drug metabolites. And when we talk about ADME in with respect to the drug discoveries, this is one part where metabolites are very important as a study. Well, probably some of you may not be aware what is dioxins. Dioxins are nothing but halogenated organic compounds. They are basically uh, tetrachloro dioxydibenzene, and it's the most toxic compound known on the earth. It is highly carcinogenic. It is not produced in any kind of industry. It is only produced in any kind of a thermal decomposition or thermal reactions, especially when coal is burned or any organic, any natural resources like diesel or petrol is burned. There is a possibility of producer of dioxin. So these dioxins are highly toxic. They settle on the grass, and through the grass they enter into the cattle, and when the cattle eat the grass, they get into the fats and milk. They are highly soluble in fats and not at all soluble in water, so they accumulate there, and then they can damage the genes, and that is how you can have the cancer. So uh, there could be mutation and there could be cancer production because of these dioxins, and they need to be measured in dairy products because they are soluble in fats, and especially meal, then your butter and cheese and all those products, and of course in meat. So they can be measured by GCMSMS at femtogram level. That's the kind of level one has to achieve. Okay, so sensitivity is very important in analytical chemistry. Then selectivity is important. Selectivity means how best an analyte can be selectively measured in presence of a matrix. Okay, so there has to be selectively possibility of measuring particular analyte. Example could be a simple sugar monitor in blood, which is very selective. Blood has a lot of other ingredients, matrix, but you can easily measure sugar by a sensor, and it can do it very fast. Another method could be immunoassay, which you use, and then third method could be hyphenated techniques where you can use GCMS or LCMS, and many of these techniques are highly selective. Then separation, many times you do not have methods which are possible to be used which are selective, so you need to carry out some kind of a separation, and separation could be very simple like centrifugation, protein precipitation, filtration, distillation, and many other methods. And then it could be liquid liquid extraction, it could be solid phase extraction, it could be solid phase micro extraction, and then of course there is no other technique other than chromatography as one of the best techniques for separation. So we use any of these methods for separation, and then after separation, we carry out the analysis for finding out the amount of analyte in a given matrix. Then structure is very important in many applications. It is required because it is not always what we do is to quantify the analyte. Many times we have to find out what it is, then how much it is. So you have to find out the structure. Example could be metabolite of a drug, whether metabolite is toxic or non-toxic, you have to find out. That's the part of that ADME. Then steroid consumed by a sportsman. So any sports event, the athlete or sportsman need to be tested for any consumption of any kind of a steroid by him, and if he is working under the influence of that, he cannot play, and his medal is withdrawn if he is found to be uh, using these kind of steroids. So it is very essential. So at that time, you have to definitely find out which steroid he has taken. So you have to identify what that compound is. Okay, so structural elucidation is definitely essential. When we have to find out the biomarker of a disease, we need to find out what is that, and we have to find out the structure of that. It could be metabolomic kind of work, it could be proteomic kind of work, but you have to find out a disease biomarker, and then you can look at the upregulation or downregulation of that particular biomarker to find out the stage of the disease. Then there could be another application where we try to find out active ingredient in a plant. So if there is a medicinal plant and if there is some active ingredient, you have to isolate it, you have to find out what is the structure of that, and then you have to see whether you can produce that in a lab and then manufacture that, or whether there is sufficient amount of this particular ingredient in the plant, whether you can produce or farm that plant and produce that at a larger scale. So structure is important in many applications. Then speed of analysis is very important. As early as possible, high throughput. These are the keywords in the industry. So they want to have the results as early as possible. It is not so important in research institutes or in academics, but slowly it is getting into that because they know the urgency of the research which they are doing. They have to publish their work. Otherwise, if somebody else does that work, he will lose the advantage and he will not be able to produce his work if somebody else does that work earlier than him. So the speed of analysis, high throughput is very, very important. And this high throughput is required right from sample preparation to analysis to data processing. All this has to be very fast so that the decisions can be taken quickly. These 5 S are very critical in analytical chemistry, and that is why I call them as very, very, they're very significant, and one has to understand the importance of these 5 S. The chemist has to play with these words and balance himself to give accurate and precise research. These are important, definitely, but there are some precautions to be taken while working on sophisticated instruments, and there are few errors to be suppressed. As I said, there are some errors, but before that, I would like to get into speed of analysis and talk to you about some applications where we can see the speed of analysis. This is just one example where I'm talking about fast LC, and you can see the analysis which was taking on normal HPLC 15 minutes, but by using the fast LC, it has been obtained within 2 minutes. So that's the advantage of the speed and high throughput. Here, another example where the analysis is done within 0.5 minutes. So 30 seconds, the analysis is over. And that's the kind of a speed the HPLC can achieve today. And that is why people are moving from the normal analysis to a high throughput analysis. So they can save time, they can save on power, they can save on consumption of solvent. It becomes more of a green chemistry. So all these are advantages of having a high speed. Well, this is one beautiful example which I would like to always talk about, and this is the analysis of pesticide residues in food. And there are 836 pesticides which have been analyzed in less than 10 minutes by using a LC-MSMS system. So speed of analysis and high throughput is so important. Now let us look at the errors which we can eliminate or try to eliminate to the maximum. One is sampling error. Now there are two parts in it. One is whether you have drawn your sample correctly. That means whether the sample represents the bulk, which is very important, because if the sample doesn't represent the bulk, you have had a wrong sample and that can give wrong results. So your sample has to represent the bulk, and many times we forget this, and especially when we try to analyze powders, we can have some kind of a distribution within that powder, and then if you collect a sample of a powder from one place and from the other place, there could be variation. People have forgotten to use the normal method, which is called as quartering and coning, and if one follows that critically, one can prepare a good representative sample from a lot. Okay, so this is very important. Then to prepare proper sample for introduction into the instrument. So your sample preparation also is very important, because that can add to an error. So when you are doing some kind of a liquid extraction and your extraction is not proper, what will happen? The results which you will obtain will be wrong, and your instrument may be very good, you are very good, but still you will get wrong results. So these are some of the things which are important. And as far as biological samples are concerned, they are most difficult in this regard, because they have lots of matrix, and we have to look at proper preparation of sample, and to get rid of matrix, that is very, very essential. You always get, because of this matrix, a huge background, and as the matrix becomes tough, you have more problems. With difficult matrix, one gets some background, and that is a background signal apart from the signal from the analyte. This background needs to be reduced, and it is mostly taken care while preparing the sample. So your sample preparation has to be good, and in such a way that you get rid of a matrix and you concentrate your sample. Example could be the drug in blood plasma. Then there is one more factor which is called as noise. It is an inherent part of any analytical instrument. It is always there. You cannot avoid it. This is why, and that is why it is significant while specifying sensitivity of analytical tool. We always talk about sensitivity in terms of signal to noise ratio. So for limit of detection, signal to noise ratio has to be equal to or more than 3. So when signal to noise ratio is more than 3, we can say we can detect. We cannot say we can quantify. Unless and until signal to noise ratio is more than 10 or equal to 10, we cannot do quantification properly. So there are these two limits which are very important: signal to noise ratio and limit for limit of detection, signal to noise ratio for limit of quantitation. Hence, if noise is reduced, one can get better signal to noise ratio, means better sensitivity. I'm just giving a typical sensitivity of 1 picogram of reserpine. It gives a signal to noise ratio 750,000 by LC-MS technique. So its signal to noise ratio is very high for 1 picogram of a sample. 1 picogram is something like 1,000 part of a ppb. Okay, it is so less. And just to give some idea, because I hope there are lots of students here, and they are probably, some of them are undergraduates, so they should understand the concept of ppb. Our Indian population, we consider it is about 120 or 130 billion. Okay, so when we talk about this, one person in so many 1.3 billion, so one person in this billion population is 1 ppb. So if we have to identify one person in this population, we say that it is 1 ppb. So you can imagine how low we are going down to, because we have to identify one person in 1 billion, that kind of a population. Okay, so one analyte molecule in so many analyte molecules or different matrix molecules. Okay. Well, we are coming to biology. There are several branches of biology. I'm not going into that. I tried to read, and somewhere I came across a number which is something like 65 branches of biology. Okay, so it's a huge number, and I'm not a biologist, as I said, so I have to restrict myself to some of the things which are related to bioanalysis. and I want to definitely talk about challenges in bioanalysis before we get into some aspects of uh, instrumentation in bioanalysis. The biological samples are very complex in nature. They are blood, serum, brain, tissues, urines, so many things. The analyte concentrations are mostly very, very low in these kind of matrix. What is required is to simplify the sample matrix while retaining the analytes, and that is why what we do when we do a good sample preparation. The key is to have a selective extract the analyte and of the analyte and concentrate it while removing the rest of the matrix component. So if we can achieve that, if we can concentrate the analyte by removing the matrix, then we will be able to do the measurements of the analyte at very, very low level. The biological research is centered around on the identification and quantification of different analytes from small molecules, which could be drug, pesticides, secondary plant metabolites, metallic elements, etc., to proteins, to subcellular structures, and so on. Okay, so it's a very, very challenging matrix we have, and what we are trying to determine is very small thing in such a huge matrix. Well, we come to some kind of a sample preparation, some general guidelines when we want to prepare sample. This is with respect to the matrix and analyte. So I have made 4 parts of whatever sample we have. And these 4 parts, I mean, all the kind of samples which we generally come across, they can be classified into one of these 4 parts. Okay, so there is a matrix and there is analyte. So what we measure is analyte or analytes, and rest of it is called as matrix. So there could be one part which is inorganic analyte and inorganic matrix. When we have such a kind of a composition, when we have to measure something inorganic and in inorganic matrix, generally what we do is a method of preparing of a sample is acid digestion on hot plate or by using a microwave digestion. Okay, so there is a microwave oven like what we have in domestic microwave oven, but more specific for the laboratory purpose, and in which we can do the acid digestion. It could be closed vessel or it could be open vessel digestion. Generally, we prefer to have a closed vessel digestion because many times we could have elements which could be volatile like arsenic, mercury. So these elements being volatile, if we do it in an open vessel, the elements will vaporize, and if we want to measure them, we have to have a closed vessel digestion. Then closed vessel gives you additional advantage. The closed vessel digestion, that system or that vessel which is closed vessel digestion vessel, which acts like a cooker. So what happens, the pressure inside increases as you heat that. The boiling point of acid, because this is acid digestion, the boiling point of acid will increase because of the pressure of acid which is created, and the boiling point of acid can increase by more than 100 degrees centigrade. So you can have a boiling point of hydrochloric acid instead of 110 degrees or 120 degrees going up to 210 degrees or something like that. And then because of that high temperature, the dissolution of the analyte will be very fast and you can achieve the speed of analysis because the sample preparation time will get reduced. So acid digestion in a microwave oven is more preferred many times. And then when we have to analyze inorganic analyte, the techniques in hand which we have, which can do trace analysis, are atomic absorption spectroscopy, inductively coupled plasma atomic emission spectroscopy, or the ultimate is ICP-MS kind of a technique, which can do this analysis very well. An example could be Bhasma, inorganic reagents or catalysts. So impurities in Bhasma, say Tamra Bhasma will have several impurities and Low Bhasma will have several impurities, and those can be analyzed by using the this particular acid digestion system, and this comes under inorganic analyte in inorganic matrix. We can have inorganic analyte in organic matrix, where we can do acid digestion, because again we have to determine inorganic analytes, so they have to be in solution and they generally will dissolve in hydrochloric acid or nitric acid, so we have to dissolve them in that. And then because organic matrix is there, we can use one more technique apart from digestion, we can do ashing of that organic. So when organic will burn, what will happen? The ash will form, and ash is nothing but inorganic. And after forming ash, we can then digest it in acid and then use it. Okay, so there are two techniques available. One, you can do direct acid digestion and use a oxidizing acid like nitric acid or perchloric acid, which will oxidize the matrix fast, organic matrix fast. It will get converted to CO2, nitrogen oxides and water, etc., and will go out. And what will remain is the inorganic elements dissolved in acid. And again, we can use the same techniques like atomic absorption, ICP, ICP-MS, etc. And then example could be toxic elements in food, blood. Many a times we come across that arsenic has to be analyzed in food. Now FSSAI, where we have that food uh, analysis tag is there, which guarantees you the safety of the food. There we have to determine these toxic elements in food. Okay, so if there is a contamination of any of these elements in human, you have to measure the contents of these toxic elements in blood, or even in skin, or even in nail, or even in hair, because many times these toxic elements accumulate there. Okay, so that's one part. So when we have inorganic analyte, these are the methods to be used. When we have organic analyte and inorganic matrix, now when organic analyte has to be taken, we have to have an organic solvent for that, because organic analyte will dissolve in organic solvents. So we use liquid extraction or solid phase extraction or solid phase micro extraction, etc. And then after taking this analyte into organic solvent, then we can use, again, for trace analysis, we can use GCLC, or if it is really very low level of detections, then we go for GCMS or LCMS, depending upon the quality of the analyte which we want to measure, whether it is volatile or non-volatile, etc. And the example could be inorganic matrix, so it could be soil and pesticides in soil. Pesticides are organic, so we can measure them. So this could be one example. Then we have organic analyte in organic matrix. Again, we have organic analyte, so it has to get dissolved into organic solvents. So we again use similar methods. We again use similar methods for analysis, but the sample could be drugs in plasma, pesticides in food, where we have analyte which is organic and the matrix also which is organic. So most of the samples can be categorized into these four areas, and roughly you can decide which techniques can be used when you have this kind of a challenge in front of you. Okay. Well, these most of the analytical instruments got developed for the reason of the development which has happened in pharmaceutical industry, then proteomic research fields. So that has compelled scientists or instrument manufacturers to develop better and better instruments. And these developments definitely has helped in many other areas. And we are able to take advantage of all these instruments into several other areas and in academic research also, because these instruments are definitely very useful. So we have to definitely use these instruments in our research, and we have to definitely take benefits out of these instruments for the quality research and quality publications when we produce our papers for publications. Well, I'm talking about all different techniques, applications, and limitations of biological analysis in analytical chemistry. So here I am taking cell and tissues as one of the matrix, and then what are the instruments which are possible to be used for small molecules as well as for large molecules like proteins. So you can see there are lots of instruments which are listed there when we talk about proteins, and the first instrument of course is mass spectrometry. Then there are other like there is histochemistry and immunostaining, then gel electrophoresis, fluorescent labeling, enzyme activity, then isotope BCA assay, non-specific. Then there are possibilities like isotope specific detection by MS. Then quantitative analysis is possible, targeted analysis, targeted proteomics is possible. So if there is a protein biomarker, we can find out the quantity of that protein biomarker. Then limitations are there because the matrix is difficult. So complex purification and sample preparation is required. So you will again see the sample preparation part is coming everywhere, and that's the critical point in any analysis. Then second thing is large and complex data sets are generated because of there is a proteins. So large amount of proteins are there in any given matrix, and when you want to analyze these proteins, large data gets generated, and this data has to be also analyzed very quickly. When you come to small molecules, then again, especially with respect to metabolites, drug metabolites, you can again find mass spectrometry is playing a major role there. Then you can use X-ray fluorescence for elemental analysis. Then you can use different fluorescent probes which are very specific. Then you can use NMR for structural elucidation. Then you can use isotope specific detection by using carbon-13 or any other nitrogen-15 kind of stable isotopes. In some cases, you can use radioactive tracers also for finding out, but then you have to use not mass spectrometry but other equipments. You require to do quantitation. You have to do targeted analysis for a particular specific compound of interest. Then in vivo analysis has to be done. Spatial identification needs to be done, and then always limitations are low analyte concentrations. When low analyte concentration may not be a problem as such, but low analyte concentration is difficult matrix is a problem. So that's one of the things which one has to determine. Then another thing is potential exogenous effect that comes because of the matrix. So that many times, whatever method you are using, especially when we talk about mass spectrometry, there is a possibility that these matrix can suppress the ionization, and whatever sensitivity you can achieve by normal way, if that matrix is not there, you cannot even reach anywhere near to that because of this matrix effects. Then there are clinical applications. So again, you will find mass spectrometry is on the top. Then you can do some assays. Then histochemistry and immunostaining is again there. You can use simple instrument like UV visible spectroscopy. And then there is absolute quantification is possible. High throughput is possible because if it is a clinical kind of work, you want to have results as early as possible. You can see what kind of problems are being faced for when we try to do a testing of COVID-19. It takes more time because the methods are not really, it is so sudden that methods could not be really developed, which could be very fast. Direct implications for health outcomes, because these tests are very, very important. Limitations, there are lengthy method of validation, and that's the problem because one has to validate any method. So if anybody develops a method, it has to be properly validated before it is put into practice. Then there is another way where we are doing a lot of structural analysis. As I put, one S stands for structure, and again, it could be molecular, cellular, everything. So imaging mass spectrometry is something which is coming up. Probably I will show you one slide on imaging mass spectrometry. Then FTIR can be used for structural elucidation, electron microscope, X-ray crystallography. Single crystal X-ray can give you some structural information about protein, three-dimensional structure of protein. Then possibilities are again isotope specific detection by using mass. So you can have a stable isotopes. So you can do in vivo analysis even for animals directly and even for human beings, because the stable isotopes are not radioactive, so you can use them very well. Then you can determine nanoscale structures by electron microscope. Limitation, extensive sample preparation. Again, sample preparation is coming there for especially electron microscopy and crystallography. Then potential exogenous effect. Again, this is coming, the matrix interferes, and need for synchrotron radiation crystallography. I will talk a little bit about that, and this is a new technology which is coming up. Well, this is what multi-tissue imaging. So I just wanted to show you what is imaging. So this is multi everybody from biology generally have heard the word MALDI, which stands for matrix assisted laser desorption ionization, and it is generally used with time of flight mass analyzer. So here also it is used with that, but in addition to that there is a imaging which is done. So this is a human tongue, and the human tongue has been analyzed by taking the sample and then uh, analyzed different proteins by using a MALDI top, by using a matrix like cinnamic acid or synapinic acid. And then you can see the white patch there, which is a defective patch, and that's the place where there is some kind of a different disease cells are there, and such kind of analysis is possible by using MALDI tissue imaging. This is again on a tissue digestion. So even on a tissue, you can do a trypsin digestion, and then you can look at the peptides which are formed from the protein, and you can analyze the digested sample on the tissue itself. Okay, so this is eczema is a MALDI-TOF instrument, and that can be used for the analysis like that. You can get a tissue imaging followed by even a spectra, and you can use a peptide mass fingerprinting to find out the protein structure, etc. Well, mass spectrometer and many other equipments are used for omics, and many of you are familiar with genomics or transcriptomics or proteomics, metabolomics, lipidomics. Today I'm not going to talk on any one of them. I'm going to talk about one more omics which is coming up, which is called as metallomics. Okay, this is a new omics which is coming up. Probably many of you must not have heard about this. This is a new thing because of the reason that there are about 30% of proteins which are metal ions, and how they behave has to be really tested, and you require a special equipment for that. And then there are 50% of all enzymes require metal ions for their functioning. And that is why this new omics science shares with its peers like other omics an unwavering reliance on advancing analytical technology to deliver results. So this is a new technique which will give you a lot of additional information. In what way? We'll have a look at it as we proceed. Well, there are two lines of inquiry with respect to omics and system biology. One is the question which we can ask: how can analytical chemistry be improved to better answer key biological questions? And are the right questions being asked that take advantage of the tools at our disposal? So if we can answer these two questions, we will be able to solve many problems. And for that, there is a good knowledge of analytical chemistry is required, there is a good knowledge of biological sciences is required, or there have to be two good people who will be sitting together and trying to look at the problem which one is facing, understanding the both the things properly, and then try to sort out the issues. That has to be done. Generally, we use the word logically. One can think very logically, but in science, many times logic can fail. But I use a specific word today. It is called as analogic. So you have to have an analytical logic. So analytical logic is more important. And if you can think with the angle of analytical chemistry and think logically also, combining these two aspects, understanding the biological aspects of a research, probably then you will be able to sort out many of the problems related to omics. So this is what we call as metallomics. And which are the tools? Because these are metal elements which we are talking about. There are tools like ICP-MS, because ICP-MS can give you the elements from periodic table, a lot of elements, more than 75 elements it can analyze from the periodic table. So almost from element number 6 to element number 92, except a few elements in between which are your rare gases, helium, argon, neon, etc. Those cannot be analyzed, while most of the other elements are possible to be analyzed at different sensitivities. Maybe nitrogen, carbon, these elements at very, very detection limits are not really very good. but they can be measured. There is new technique which I am going to talk a little bit, one slide only, which is called as synchrotron XRF, and that's the one which is good technique for this metallomics analysis. Why it is so? We'll see why it is so. So the basic reason why it is used is to give you oxidation state of metal in biological sample. So the other techniques which I have described above, ICP-MS or wavelength dispersive XRF or energy dispersive XRF, they can give you total metal concentration. So when you try to measure iron, Fe, it will just give you total Fe. It cannot say whether Fe is in divalent or Fe is in trivalent state. Now, there are many metals which can have multiple oxidation states, and the oxidation states cannot be measured by these two equipments. For that, you require some new equipment which is called as synchrotron XRF. Well, we have to first understand the available instruments as on today, and we'll try to look at this atomic spectroscopy to some extent. Again, I'm looking at the entire audience and trying to match everybody so that everybody would be able to take something out of whatever I'm giving a presentation about. Okay. So let us talk about these three techniques: ICP, ICP AES or OES, and ICP-MS. So in these techniques, what we do is we prepare the sample in the form of their acid salts, that means their chlorides or nitrates. And when these are aspirated at high temperature in a flame or plasma, they will get atomized, they will get vaporized, they will get atomized, and these atoms can be used for characterization. So there are three ways we can talk about. When we talk about atomic absorption, the atomization is done in the flame. Of course, you have a sample prepared, solution is aspirated into the flame. Flame will decompose the molecule and it will form individual atoms. And if the temperature of the flame is around between say 2000 to 3000, most of the elements form atoms, and these atoms are at the ground state. And when these atoms absorb the radiation coming from a source like halocathode lamp of a particular emission line, which is characteristic of the element which we are trying to analyze, the electron from the lower level goes to the higher level. From the ground level, it will go to the higher level by absorbing that particular radiation. And we look at the absorption of the radiation by atom which is at ground state, and then we call this technique as atomic absorption, for which we require external source of lamp which we call as hollow cathode lamp. These can determine elements, something like 30 elements can be easily determined, but the sensitivity of this is not very high. And then it is not a qualitative analysis tool. If an element is present, you can measure it, provided its detection is matching with the capability of the atomic absorption spectroscopy. There are two types of atomic absorption spectroscopy equipments. One is based on flame, other is based on graphite furnace. Graphite furnace has a better sensitivity compared to flame. As I'm not going to talk on this technology as such, I'm not going into those details, but remember that atomic absorption is a quantitative analysis tool. It is not a qualitative analysis tool. Then we come to atomic emission, where we use a very high temperature. The temperature in atomic absorption doesn't exceed 3000 generally. I'm talking in general. While in atomic emission, when we have a plasma, argon plasma, the temperature of this plasma can be above 6000 up to about 10,000 degrees centigrade. At that temperature, most of the elements not only get converted into atom, they get excited because of that high temperature. So most of the elements are at this stage in atomic emission. They are not at this stage. So what happens when they are at this stage? They come back to the ground state by releasing energy. And this energy, which it is released in form of radiation, which is proportional or which is responsible for a particular element. So we get a particular emission line. This could be for, say, copper. Another element will give another emission line. with other different wavelength. So when we use atomic emission spectroscopy in this format, analyzing number of lines which are coming at different wavelengths, we can find out which element is present. So by atomic emission spectroscopy, which is called as ICP-AES, that is inductively coupled plasma atomic emission spectroscopy, we can determine more than 70 elements. And this is done very fast because of the present detectors which we have. We can analyze 70 elements very fast. While in case of atomic absorption, you can analyze one element at a time. So the time taken for the analysis is very high, while in atomic emission, ICP AES, the time taken for the analysis is very less. Okay, so we can do number of samples in a day. While when we have a mass spectrometry, we use exactly same technology. We have a ICP-MS, so we use the plasma, which is argon plasma, and then, as I said, most of the elements go to the excited state, and also most of the elements will lose electron, and they will also get excited so much that the electron is lost permanently from this atom, so it becomes a positively charged ion. And if it becomes a positively charged ion, it can be put into mass spectrometer, because mass spectrometer can separate ions on the basis of mass to charge ratio. So once the electron is excited and it goes out, then the atom becomes a positively charged atom, and then this can be easily measured by using a mass spectrometer. So ICP-MS is just nothing but a similar ICP, but now instead of measuring the emission lines, the photons or the radiation, electromagnetic radiation which is coming out, we look at the ions which are coming out, focus them into mass analyzer, and then mass analyzer is able to separate those ions on the basis of mass to charge, and you can measure them. Okay, so these are three techniques which I have tried to explain in one slide, and probably you will be able to understand. So this is again the same thing. So you can see here we have a halocathode lamp, a flame, you have a monochromator which will separate the radiation, and you have a detector which will sense one particular element at one time. Well, when we have ICP, we don't require a source of a lamp, because sample itself acts as a source, because in any spectroscopy you require a source of radiation. Your sample itself acts as a source. So different emission lines which come, it becomes a polychromatic radiation, because there are different elements present in your sample. All of them will give a polychromatic radiation, which is diffracted by using a monochromator, and all the elements are put onto a CCD detector. So there is a XY plane detector, and each diode on this detector is for a particular element or particular line of a particular element, because every element gives you several emission lines. So you can get image or the signal over here, which is amplified, and you get at the moment or at a given moment, you will be able to see all the elements which are present, and you can qualitatively find out how many elements are there, what are those, and then depending upon the intensity of the element, you can find out the concentration of each one of them. So it becomes a ICP-AES, so you're looking at the emission lines which are coming. Now the same plasma here, what is done here, I'm not looking at the emission lines here, I'm looking at the ions which are created in the same plasma. By losing an electron, positive ions are created. They are pushed into mass spectrometer, and mass spectrometer will separate the ions on the basis of mass to charge ratio, and these ions will get detected, and you will again be able to see, depending upon the mass of individual ions, we can find out which element is present, and depending upon the intensity of the radiation, intensity of that ion, you can find out how much is present. So both these equipments can do both qualitative and quantitative analysis, while the atomic absorption can do only quantitative analysis. So this is your ICP-MS, which gives you, this is the plasma, this is the quadrupole mass analyzer, which is the detector, and you can go down to very, very low level, parts per trillion level. Okay, so there are strength of this, excellent detection limits. It can do a lot of elements together. economical for many samples, and excellent screening tool, so you can find out what it is there. You require some method development skills, so good analytical skills are required. Higher initial capital costs, so costly, and some spectral interferences can come because one element can give you several lines. So there could be several ions which have got interferences, and those ions, because if they have same mass, they will come at the same time, and because of the isotopes, there is a possibility of one isotope can match with another elements, the main abundant mass of that, and then that can interfere. So you have to have interferences which need to be removed, and it has low TDS. So dissolved solids can create a problem. So you should have low dissolved solids. This is how the plasma is produced. I don't know whether I have time. Yeah, I can still go ahead. And this is where you have a argon gas which is coming like this, and this is the induction coil to which RF voltage is applied. So very high RF field is generated. This is a copper coil. So this copper coil to which RF field is applied, and there are three quartz tubes, one inside the other. Through one, argon gas flows tangentially. Through the central one, your sample flows along with argon gas. So argon is coming, and its sample comes along with that by using either peristatic pump, and it comes into the plasma, which is here. And there is one more gas which has which has to do one important job, to lift the plasma a little higher so that the plasma doesn't touch the quartz tube. Otherwise, quartz will melt, because as I said, temperature of plasma is above 6,000 degrees centigrade. No quartz can withstand that temperature, it will melt. So you have to lift the plasma up. So the pressure of argon lifts the plasma up. So one argon is here, argon is inert gas. So you have to seed the ions into that. So there is something called a Tesla coil, to which you apply a very high potential. So it gives a spark, and because of that, the argon atoms which are there in this area get ionized, and once they ionize, the RF field will work on them, and the ions will start going towards positive and negative. When the coil becomes positive, they will get repelled. When coil becomes negative, they will get attracted towards it. And because of this continuous movement of argon at the RF voltage is very, very high. It is 28 megahertz, and that's too high. So the movement of this argon makes it thermally heated, and it generates a very high temperature, which gets converted into argon plasma. Plasma is nothing but the excited atoms, the positive ions of that, electrons. It is a mixture of all of them, and we call it as generally fourth state of matter. And this argon plasma has a very high temperature, as I said. Okay, these are all the elements which are possible. So you can see what are all the elements which are possible so all the elements which are listed here, except the one which are given by this particular color, all the other elements are possible to be measured by ICP-MS, of course, except these actinide series elements which are radioactive. So this is how the mass analyzer works. So this is a quadrupole mass analyzer which selects one ion to go to the detector at one time, and at that time all other ions are discharged, and only one ion gets stabilized and go to the detector by changing a DCRF voltage. So this quadrupole, we apply a DCRF voltage. At one DCRF voltage, one ion gets stabilized and go to the detector. As you change the DCRF voltage, maybe at next DCRF voltage, this blue ion will go to the detector and the other ions will go out. So one by one, you will be able to detect all the ions which are coming. So generally, we know what elements we are trying to measure. We jump from that mass to the other mass, and then try to measure a particular element of our interest, and go to the next element and go to the next element and measure all of them. So initially we do qualitative analysis and then do quantitative analysis depending upon which elements we have to measure accurately at very low level. So this is how you have a plasma here getting generated. There are two cones which will allow the ions to get in. The neutrals and other things are eliminated. It goes to the what we call as a collision cell, where we try to remove the molecular interferences. As I said, I will just give one example quickly. Now, Fe has a mass of 56. So if I want to measure a Fe ion, its mass is 56. Now, argon and oxygen at the temperature of plasma, which is much higher, argon and oxygen can combine. So argon is 40, oxygen is 16, to form a molecular ion which is called as 56 again. And both of them now are in 56. Ion is 56, argon oxygen molecular ion which gets formed also is 56. So this collision cell gets rid of this argon oxygen molecular ion by using energy discrimination. So we apply a helium gas here. It collides with a large argon oxygen molecule and decrease its energy. So it goes into a different direction. It will not go straight, while Fe will be able to go straight and can be measured by the detector. And that is how we require some device which is called as collision cell to remove the interferences which are coming, which need to be removed. So this is collision cell. I'm not going into details because that is not the purpose. This is how the ICP-MS look. This is the quadrupole, there is a collision cell, and the plasma is outside. Yeah, I'm not going into details. Okay. So this is some result which is for the diagnostic. So we have analyzed some sample for blood for arsenic, cadmium, lead, mercury. Most of these are toxic elements, and one sample which was analyzed has some amount of lead in that. All others are not detected, so they are much less. So this is just some report which I collected. I'm giving some real life examples and trying to convey whatever I want to say. This is cisplatin. This is a drug which is used for cancer treatment, ovarian cancer and many other cancer, even. Yeah, so it is used for cancer treatment. And this has got a platinum. And this platinum has to be determined. And this, of course, has some adverse effect because of it is used as a chemotherapy agent. So cisplatin, it is also called as carboplatin, and it has very high toxicity also. And it is about 50 to 100 milligram every 3-4 weeks has to be given. It has a lot of side effects, and one has to determine its presence in the blood cells. So it is supposed to act like that. So generally this has got platinum has valence state. It can have a 2 or 4. Here it is having chlorine on one side, 2 chlorines, but now here we are seeing they are attached to the amino acids here, and they have created whatever cell DNA is there that has been attacked by this, and that is how the replication of that can be stopped, and this is how it controls the cancer. That is what is expected. So there is another one which is arsenic toxicity. So arsenic is the element which is again highly toxic. So we talk about one aspect of using cisplatin as a drug for chemotherapy, and we look at how this acts inside the cell. By using a ICP or ICP-MS kind of a technique, we cannot really find out what is the valence state of platinum, and we know total platinum. So if we measure platinum in the patient sample, we'll know the total platinum. We will not know whether it is in +2 state, +3 state, +4 state. We will not be able to know, and that's one of the drawbacks. This is again arsenic, which is also present in various valence state, and we have to find out what state it is. So there are examples where the water quality is bad, especially in Orissa and Bengal. There is some areas where water is found out in groundwater, especially has a lot of arsenic in that, and that creates a lot of problem. You can see it comes onto the skin, and skin gets totally damaged. I have a lot of other photos which are not put. The source of ACE, it could be organic ACE found in water, or it could be elemental ACE in some valence state. It is found in apple juice, orange juice, milk, dairy products, cereals, and beef as inorganic. Organic arsenic is found in water, in soil, grains planted in such a soil. Organic ACE is less toxic and inorganic is more toxic, and it generally arsenate as 5 valence state or arsenite as 3 valence state, and it gets accumulated in skin, nails, and hair. So it has a lot of effects because of that. Arsenic induced cardiovascular dysfunction, arsenic induced diabetes, arsenic induced neurotoxicity, arsenic induced nephrotoxicity, arsenic induced carcinogenicity. So there are lots of problems which can be created by arsenic. And then if we have to determine this arsenic into the tissues or something like that, we can use some technique like laser ablation and take it out from the tissue. So laser is heated on this target, and because of the laser heat, the arsenic gets vaporized, and then it can be collected and sent to the ICP-MS, and you can analyze arsenic. So there is a LA, there is ICP, and there is ICP-MS, and you can find out the arsenic content in by using a LA-ICP-MS. There is some technique which is available to find out a state of arsenic, whether it is arsenite ACE3 plus or ACE5 plus. You can have also, in addition to anything else, if you can take it into solution and retain the valence state, you can use ion exchange chromatography, and you will separate ACE3 and ACE5 and then push them in ICP MS, and then you can determine total arsenic here. But at a particular retention time, say arsenic 3 will come, at another retention time arsenic 5 will come, and then you can find out the valence state of arsenic 3 or arsenic 5 by using ion exchange chromatography coupled to ICP coupled to MS. Okay, so that's one way. If you want to analyze directly on a tissue, you can use a technique like laser ablation, but it will again give you total arsenic. Well, this is a element present in brain, and this is of course a rat brain which has been analyzed for various elements, and as you can see, carbon 13 is analyzed, magnesium 24, phosphorus 31, manganese 55, copper 63, Fe 56, and zinc 66. So here, of course, these blue ones are your Fe and copper 63. Okay. So, well, I come to the last slide of my presentation probably. And this is a new technique which is going to be very useful for that new metallomics, because this technique can give you within the cell what is the valence state of a particular element. And many times this is very important, because this valence state of a metal can form a various type of complex with the protein or the chemicals within the cell, and it is very essential to know what is the valence state of that element. So whether it is going to get oxidized or whether it is going to get reduced in the reactions within the cell, that has to be measured. So this synchrotron X-ray fluorescence microscopy gives you image. It can give you microanalytical technique for the quantitative mapping of elemental distributions. Among currently available imaging modalities, XRF is the only technique that is compatible with fully hydrated biological samples such as whole cells, tissue sections, etc., while simultaneously offering trace element sensitivity and submicron spatial resolution, combined with the ability to provide information regarding the oxidation state and coordination environment of a metal cation. XRF is ideally suited to study the intracellular distribution and speciation of trace elements, toxic heavy metals, and therapeutic or diagnostic metal complexes. So this is a technique which is coming up. This will revolutionize the modern concepts in analytical chemistry with respect to biological sciences. So The conclusion, it is very necessary that analytical chemists and biologists engaged in sustained dialogue so that both fields continue to flourish. Okay. So there is a need of good analytical chemist for a biological science, or biological scientists should know the good aspects of analytical chemistry. Probably with that in mind, Dr. Sane sir has started this new MSc course which is called as bioanalytical chemistry. But then there is definitely a combination of both these fields are required. And you can see the health science is going to be current priority considering the present circumstances COVID-19. Though the gap between analytical development and biosciences developed is wide, trends are indicative of possible shift, so that we should all have our common goal, the pursuit of knowledge. Fine, thank you all being here in such a large number. Thank you to Biological Science Department of GN Khalsa College team for organizing this lecture, and all the best to all of you. During this time, you are spending some crucial time for this kind of understanding of new concepts in various scientific field and enriching yourself. I hope I have given you some inputs in a short time on a very vast subject. Thank you so much again. All the best to you. I conclude.
Thank you, sir.
Last slide.
Uh,
This is with respect to COVID-19 and the present situation. This is a thought of Marie Curie, who is no more, and this particularly a sentence which she had probably written with respect to radioactivity. Well, it is applicable in the present situation. Nothing in life is to be feared. It is only to be understood. Now is the time to understand more so that we may fear less. Okay. So with that, I conclude. Okay. Sorry for this interruption, Dr. Nomita.
Absolutely not, sir. So moving on to question answers.
Yeah.
Uh, is dioxin detected in milk and milk products in our country?
Yeah, it is done today. There are two places this particular instruments are available. There are two types of instruments, as I said. There is a high resolution magnetic sector mass analyzer connected to GC, and this
Okay.
facility is it Export Inspection Authority in Chennai, and it is also with Vimta Labs in Hyderabad. These two
Okay.
facilities I know very well, and people are now trying to use it by using the GC-MSMS, quadruple GC-MSMS technique. And we have, as a Shimadzu, we have this method available with us. So if anybody is interested in such kind of measurements of dioxins and furans, there are two types of toxic compound, dioxins and furans. And these are all halogenated dioxydibenzene, and then it is a monoxy furan kind of a compound. So tetrachloro. So all of them have a chlorine in that. And there are several isomers of all these, and they can be measured by using these two types of instruments. And
Okay.
these are available in India.
Okay. Prachi Pandya asks, if there is no peak area in LCMS, how can we conclude our result, especially for unknown plant sample?
Yeah. Can you repeat? You have no?
No peak area in LCMS for unknown sample. How
Yeah.
do we conclude the result?
No, generally, when you are doing something, you cannot call it as a known. Okay.
Yes.
Yeah, because there are two things which are known.
Hmm.
One thing is the solubility of your sample.
Hmm.
Okay, if your sample is soluble in polar solvents like methanol, then generally it can get ionized by LCMS in LCMS, and you can get it measured. It is possible. Okay, so
Yes.
rough thumb rule like that. So the
Hmm.
polarity of the sample is very important. Okay, so
Okay.
if it's polar in nature, and if it is soluble in water or methanol, or even to some extent acetonitride, you can get it ionized in LCMS, and you will be able to see the peak area or peak. I don't consider a peak area, but the peak needs to be seen. If you get the peak, you know definitely then the mass of that compound, and
Hmm.
doing MSMS or fragmentation of that, you will know roughly some idea about what that compound could be. Okay,
Right.
so you can get some good information provided you do a proper work. What is happening is most of the time all these people who do the extraction give the
Hmm.
sample to some organization or some institute where they have a LCMS-MS or LCMS,
Hmm.
and the operator who works on that will just put the sample into whatever column he has and whatever condition he has already set up, and will give some results. And many times those results are absolutely useless. They have
Right.
wasted the time and money because they have to pay for this analysis, and they
Hmm.
do not get good results at all. So
Hmm.
you first discuss this kind of samples with somebody who understands mass spectrometry very well, and then give the samples with proper instructions to the operator who is operating the machine. Remember,
Okay.
most of the institutes have operator. They
Hmm.
don't scientists to work on that machine.
Hmm. Hmm. Yes.
Operators will only operate the machine as per the instructions or SOP which is given, and that SOP may not be valid for your sample.
Hmm. Hmm.
Okay, so
Right.
problems come because of that.
Okay. So Dr. Mangesh Dagawal asks, if we
Hmm.
have only a plant powder and we want
Hmm.
to know a specific compound from that, how do
Hmm.
we go about it, right from basic to advanced?
Okay, now the plant powder, again, no, you have to find out what you are expecting in that. Now there is some literature available about every plant,
Hmm.
and if you have collected a plant powder from which part of the plant, then
Hmm.
what is the solubility of that powder?
Yes.
To be some solubility, at least partially it would be soluble in some solvent, partially it would be soluble in some other solvent, and from the polarity of the solvent, you know what you are trying to use or what you are trying to look at. Are you
Hmm.
going to look at the polar compounds, or are you going to look at some non-polar compounds?
Hmm.
So
Hmm.
you have to work yourself on that powder in such a way that you will be able to get some information. You can use a simple technique like TLC. I mean, if it is partially soluble in some solvent, you can use TLC because that's a good technique. If you use a solution which contains powder on HPLC, it will damage your HPLC. But when you
Right.
put it on TLC, if there are some suspended particles, they will settle where you put your sample, and then whatever bands you get, depending upon the mobile phase which you use for uh, pushing that sample up onto the plate, you will
Hmm.
know what kind of polarity that compound has. And then taking those bands which are uh, there on the TLC plate, you can extract and you can try to analyze them separately by LCMS-MS technique. Okay, so there is a way of analyzing everything. Okay,
Right.
uh, yeah.
Okay. Mr. Siddharth Sagar, I have
Mhm.
performed Orbitrap LCMS-MS for brain
Mhm.
tissue lysate proteins, and
Mhm.
post that he has done HR LCMS-MS for band protein separated on native gel. How
Mhm.
should he correlate these two techniques?
Okay. Now, does he try to analyze this, whatever information he's getting, which is very complex, by using any bioinformatics tool?
Hmm.
He knows which proteins he's analyzing,
Hmm.
source of proteins which is analyzing, whether he could get any information from the data which is available,
Hmm.
any informatic tool where databases are there for similar kind of proteins, whether he could get some information from there. So he has to look into those aspects because he probably has collected huge amount of data. Now that needs to be properly analyzed. I'm not a protein chemist or
Right.
kind of a person, but if he really talks to someone who has a good background of like I can give you suggest some names like Dr. Mahesh Kulkarni from National Chemical Laboratory Pune, or
Okay.
there are some good people in ISER Pune. They can definitely guide you on this.
Okay. Isha Savant asks, will reactive oxygen species not react with isotope carbon 13 detection?
Mm, reactive oxygen species.
Hmm. Will it not react with isotope carbon 13 detection?
Where?
That is
Where
not
it can
mentioned, sir.
react?
Which technique,
Outside
what
or
way?
inside? Where
Not mentioned.
your
Hmm.
reactive oxygen? What do you mean by reactive oxygen? And why
Hmm.
carbon 12 or carbon 13?
Okay.
Yeah,
Next.
it doesn't happen in mass spectrometry, definitely.
Hmm.
If it happens in ionization source, also it is not possible. So any kind of such interactions are very rare in mass spectrometry. I do not know
Okay.
where she's talking about such kind of reactions.
Yes. So I'll just take last two questions. Which one is the useful method for protein, specifically enzyme molecular structural analysis?
Uh, Yeah, if for proteins, one can use definitely LCMS or LCMSMS, especially LCMSMS, or initially to get a multi-top for intact protein analysis, then do a trypsin digestion to get information about the peptides which get formed because of the trypsin, and then try to analyze those trypsins, digested protein by using LCMSMS. And then also if he can have a proper sample preparation and protein purification is possible, he can prepare a single crystal of that protein, then he can use a single crystal XRD for very good analysis of the structure of a protein. But it's more complicated. Sample preparation is too stringent there, so he can take off the people who are working on single crystal XRD, and there are places where it is already there. It is there in Indian Institute of Science, Bangalore. It is there in IICT, Hyderabad. I'm not very sure it is there in IISER, Pune or NCL. I think NCL should have it. So he should be able to get, if he can purify his protein and do this kind of analysis. Otherwise, the technique like LCMS or LCMSMS would be able to give you at least the complete chain of peptides, and he can assemble the structure of peptides. Of course, again using a database, proper database for that protein.
Okay. So last question.
Yeah.
Rishiraj Gupta asks whether ICP-MS can differentiate between dissolved and suspended metal in wastewater.
No,
Okay.
it can only it be used for dissolved. Okay,
Okay.
if it is suspended particles, then it is not possible. Okay,
Okay.
because they may not get uh, into ion formation, and they can create a problem in the nebulizer of the ICP-MS.
Okay. So there are many more questions which we will mail you,
Mm-hmm.
and maybe we can reply to the participants again.
One more from Nidhi actually.
Okay.
Okay.
So she wants to know whether there are any analytical techniques available that help
Hmm.
differentiate bacterial strains and mutated types from wild types.
Arey baap. No. Probably I'm not a good person to answer this question, to be frank, because I have never done this kind of a work in my life. Staining the no. This is sorry for that. I'm very sorry for that, because as I said, I'm not a biological chemist, so I may not be able to really answer your question straight away. I probably may be able to do that after reading something or after discussing with some of my friends in this regard.
So that would be wonderful, sir. Yes, sir.
Yeah.
Thank you. Yeah.
Maria, could you please give the instructions?
I think Ketki can proceed.
Yes. Ketki, vote of thanks, please.
Hello, good evening, everyone. Today I am highly delighted to deliver the word of thanks to our eminent speaker, Dr. Ajit Datar sir. It is said that nothing is as important as passion. No matter what you want to do with your life, always be passionate. One such passionate teacher we all know is none other than Dr. Ajit Datar sir. On the behalf of the Department of MSc Bioanalytical Sciences, Guru Nanak Khalsa College, I sincerely thank Dr. Ajit Datar sir for highlighting the importance of modern analytical instruments and its application in biological sciences. So you helped us to understand how biological samples are highly complex in nature and how these techniques help in structural elucidation and identification of the biological markers, which are of the utmost importance in disease identification and drug discovery. Today, we were even aware about certain hyphenated techniques, but today you even threw a light on one of the other hyphenated technique, that is LAICP-MS. So this was a technique that even we weren't aware of about, sir. Thank you, sir, for taking out time from your busy schedule and enlightening our participants in this lockdown period, where your efforts are highly appreciated. I thank our principal, Dr. Kiran Mangaonkar, sir, for his constant support and encouragement and allowing us to conduct this webinar at national level. I thank our beloved ma'am, HOD of MSc Analytical Sciences, Dr. Naumita Dhume ma'am, for guiding and supporting us throughout the webinar. Nothing would have been possible, ma'am, without your ideas. I also thank my proactive, supporting, and dedicated colleagues, Ms. Palvi Kamath and Ms. Maria Nadar, who have given their best to make this event successful. No event would ever be conducted smoothly without an active and dedicated IT team. I sincerely thank Mr. Aldrin sir, Sham sir, and Ashish sir for constant support throughout the webinar. Last but not the least, I would also like to thank all the participants for attending the webinar in such a large number. It is said that great things happen to those who don't stop believing, trying, learning, and being grateful. I would thus end by saying, believe in yourself, think positive, keep learning, and be grateful to God for being safe in this COVID-19 pandemic. Thank you so much. So now I declare this webinar closed. But
Just before that, Etki, I would like to
Yes,
just add something.
Sure, sir.
In the next week on 26th and 28th of May, there would be two lectures. 26th, there will be a lecture between 2 to 4 on GCMS. And on 28th, there will be lecture on LCMS on 28th, same time, 2 to 4 p.m. Okay. So
Okay,
if you want, you can participate and attend these two lectures. And
sure, sir.
these lectures will be given by me on these two topics. If you are interested, please attend.
Definitely.
Thank you.
So how
Yes,
do
sir.
the participants attend
Yeah.
it? On which platform is it, sir?
Yeah, there
Can
will
you
be
share the details?
announcement. Kavita will be announcing it. Kavita will be
Okay.
putting it,
Okay.
and they will know. It will be this. I think the same platform, Gmail. Okay,
Okay.
so yeah, so they
So the
will
Okay.
know about
participants,
the
It
program
will
very
be
soon.
on Khalsa College website then.
Yeah, yeah, it is through Khalsa College only. It is.
Yeah, it will be on the
Gmail
Khalsa College
College
website.
only.
So
Yeah,
those who are interested can go through it. Yeah.
yes, they can join this.
Thank you so
Also, I would like to add something. The feedback link will be posted in the chat box and in the description box. Also, if you want to see this video of the lecture, it is available on the college website. You just have to subscribe the college channel. Thank you so much.
Aldrin, we can close the webinar.
Yeah.
Yes. Okay. Thank you.